Hi everyone, I just wanted to touch base on these recent news that you may have seen that we shared it. It's really an incredible breakthrough in science and medicine where a fully custom and personalized gene editing therapy was developed for KJ, this little baby that had or has an ultra rare and fatal genetic disorder. Gene therapy has been around in the past, but really the speed that all of this was developed is truly spectacular for industry standards, and it really shows us what what is possible today. Just for you to have a flavor of everything that went into developing this, in this period of 6 months. Obviously there was diagnosis, people had to come together, they had to develop patient cell models to test the gene editing approach. It had to be tested as well in a mouse model, and in parallel there was a toxicology study in a non-human primate, so in a monkey, to show that the gene editing approach was safe. And so all of this was done in just 6 months before the administration of the therapeutic. So, it is really a motive for celebration. And, I just hope KJ continues to improve as, he'll be a living proof of what is possible for the medical and scientific communities to accomplish when they come together with such purpose. Now, I guess one question you might be asking is, can we do this for v-ATPase disorders? And I guess the short answer is not yet, but things are moving in that direction pretty rapidly. I think there's two main challenges when developing these gene editing therapeutics. One of them is that you have to be sure that you're correcting the exact piece of DNA that is changed. And so that you have a perspective on this, we have tens of thousands of genes, and then within each gene, we have hundreds of thousands of letters or base pairs. And these CRISPR therapies must correct the exact single letter that is changed in a given individual. So you can appreciate the high level of precision that these things need to have. But again, CRISPR is allowing us to get to that level of precision. And the second challenge is you have to deliver this therapy in the cells that need to be corrected. In KJ's case, these cells were well known. They were in the liver and were cells responsible for driving the urea cycle. In the brain, it is a little more challenging. So again, so that you understand the perspective, like we have trillions of cells in our body, and somehow these things need to hit the right ones in order to see a clinical benefit. I mean, this is to say that brains are a little more challenging to target. Biology prepare them to be inaccessible to outside things, and for good reason. The main one is to protect us from infections. So most bacterial and viral infections never reach the brain because they can't. But it makes it hard for us to target the brain as well with therapeutics. But again, technology is moving really fast in that as well. So we're hopeful that in the near future we'll have more and more technologies and better approaches to target the brain as well. We also need to understand how many cells would we need to correct in order to see a clinical benefit. This is something that we'll have to study in the lab. Like, do we need to correct the entire brain? Just, is 10% of the cells enough to have a clinical benefit? We don't know. And so we need to continue doing some research and understanding this better, so that when the opportunity presents itself, that we may have a decent shot at targeting v-ATPase disorders. We're well positioned to take on that opportunity. So anyway, yeah, exciting times. This is really a motive for celebration, and to keep us all filled with hope that we may have an opportunity to cure v-ATPase disorders as well, when the time comes. So thanks, everybody.